Okay, so this is cancer immunotherapy strategies. So previously I covered the section on how the immune cells affect cancer and the types of cells, the type of immune cells. So today I'm going to be talking about the type of immunotherapies, which is generally categorized into three types: molecular therapies, cellular therapies, and vaccination therapies. So molecular therapies are subdivided into three sections. Cellular therapies is subdivided into two, and vaccination therapies are divided into prevention and therapeutics. So the first type of molecular therapies is cytokine IL-2. IL-2, also known as interleukin-2, is a signaling molecule that is found in the immune system and functions by regulating the activities of the white blood cells, which is the leukocytes and lymphocytes that are responsible for the immunity and promotion of the T cell growth. IL-2 is also capable of mediating tumor regression and was approved by the FDA for metastatic renal cell carcinoma and metastatic melanoma. It is also used to expand tumor-infiltrating lymphocytes and to support the growth and survival of the cancer cells. This molecule it also promotes a TREG proliferation and yields severe toxicities, including VLS, pulmonary edema, hypotension, and heart toxicities. The second type of molecular therapies is the immune checkpoint inhibitors. This type of drug it's a drug that blocks the protein that acts as checkpoints that are made by the immune system, and in some cases, cancer cells also produces these proteins. When these checkpoints are inhibited, then T cells can kill cancer cells easily. So the FDA approved certain drugs, these few drugs, to block the inhibitory functions and the interaction of these checkpoint proteins to reactivate T cells. Clinical efficacy is only displayed in a small portion of cancer patients, whilst most of them show resistance to these drugs. The third type is agonists of co-stimulatory receptors, inhibitors of immunosuppressive factors, and agonists of T cell metabolism. So co-stimulatory receptors are a class of molecules expressed by T lymphocytes that regulate the activation of T cells and the generation of effector T responses. Studies show that anti-4-1BB stimulates T cells, whereas anti-OX40 and anti-GITR not only stimulate T cells but also inhibit TREG. Similarly, co-stimulatory molecules CD40 on DC cells involved in DC activation. Thus, anti-CD40 has shown anti-tumor effect through its activation of DC and T cell priming. Tumor cells secrete a number of immune suppressive factors, for example, TGF-beta, PGE2, IDO, and arginase into the TME. Targeting these factors may reactivate T cells for anti-tumor immunity. Inducing defects in mitochondrial metabolism compromises TIL functions, hence targeting metabolic pathways in T cells may benefit cancer patients. The two types of cellular therapies are adoptive T cell therapy and depletion and blocking of TREG. Adoptive T cell therapies involves in vitro techniques. The antigen-specific T cells are derived from patients and are then reinfused into the patient. Adoptive transfer of T cells do not benefit just any type of cancer. It's mostly beneficial to metastatic melanoma. Depletion and blocking of TREG. TREG cells suppress abnormal or excessive immune responses to maintain immune homeostasis. These cells are involved in tumor development and progression by inhibiting anti-tumor immunity. Depletion of these cells may benefit cancer patients. The drug denileukin diftitox releases diphtheria toxin into TREG, leading to TREG depletion. TREG functions are also inhibited using antibodies against TREG markers or to block TREG trafficking to tumor sites using methyl gallate. Okay, so vaccination therapies, some cancers are caused by viruses. Example, the cervical, vaginal, vulvar, and anal cancers are caused by HPV infections, and liver cancer is caused by HBV infections. Hence, the HPV and HBV vaccines were approved by the FDA to prevent healthy people from developing these cancers. Apart from prevention, vaccines are also used as therapy. These include peptide vaccines derived from tumor antigens and vaccines derived from cancer cell DNA and RNA. Another therapeutic vaccine is oncolytic virus vaccine, such as EBV vaccine, that infects tumor cells and causes immunogenic cell death. So, not many cancer patients benefit from these immunotherapy because some of them are resistant to these drugs. Primary resistance includes low antigenic mutations in tumor cells or aberrant expression of genes in tumor cells that promote the cell adhesions or extracellular matrix remodeling. So later on, some patients who previously benefited from immunotherapies acquire resistance, and these include loss of target antigen expression in tumor cells or an increased expression of immune checkpoint proteins on tumor cells, which results in the ineffectiveness of immunotherapy. So a major approach to overcome immunotherapy drug resistance is to combine therapies. So for example, combination therapies that involve the immune checkpoint inhibitors combined with anti-CD40 inhibitors have shown to suppress melanoma. Efficacy of current immunotherapy drugs varies from patient to patient, highlighting the importance of identifying personalized biomarkers to predict who is likely to respond to a specific therapy. Cancer therapy emerges as cancer therapy, which precision, which is more precise, and yeah. It's actually more precise compared to chemotherapy and radiotherapy because it's more personalized.